You know, for literally centuries, we've dreamed of turning back the clock on aging. But what if the secret wasn't some, I don't know, complex fountain of youth, but a series of simple switches right inside our own cells? Well, today, we're going to dive into some absolutely incredible research that built a new kind of search engine, one designed to find those exact switches. So let's get into how they pulled it off.
So this whole quest to slow down or even reverse aging, I mean, it's a universal human story, right? Scientists have been tackling this for decades. But honestly, the approaches have often been super complex. We're talking really, really complicated stuff. But a new question is starting to change the whole game.
And that big question is this: what if, instead of trying to fix everything that goes wrong with aging all at once, we could find just a few powerful levers to pull? You know, single switches that could reset the whole system. Could we actually turn back the clock on ourselves with just one targeted, simple tweak? That's the idea.
Okay, so this really lays out the shift in thinking perfectly. For a while now, the gold standard for making cells young again involved this powerful cocktail of 4 proteins. You've probably heard of them, the Yamanaka factors. Now, think of that like trying to conduct an entire orchestra at once. I mean, it works, but it's complicated, it's kind of messy. The new goal? So much more elegant. It's about finding the conductor's baton, just that one single master switch that can get you a similar result.
So, to find that conductor's baton, researchers basically had to throw out the old playbook and write a new one from scratch. They built this totally revolutionary system designed for one thing and one thing only: to hunt for these master switches. You can think of it literally as a search engine for youth. And here it is. It's called the Transcriptional Rejuvenation Discovery Platform, or TRDP for short. Now, I know it looks complicated, but the idea behind it is actually brilliant. It's basically a high-speed, systematic process that just sifts through our genetic code to pinpoint the exact factors that can make an old cell act young again.
Okay, let's break this down into simple steps. First, the platform analyzes. It looks at old cells and young cells and asks, okay, what's changed? What's the genetic difference here? Second, it predicts. It uses some serious computational power to create a ranked list of factors that are most likely to reverse those changes. Then, step 3, it tests. This is the cool part. It uses CRISPR technology to go through the top candidates one by one and flips their switches on or off. And finally, identify. It figures out which of those single switches actually made the old cells look young again. Simple as that.
So I keep talking about these switches. What are they really? Well, their official name is transcription factors, or TFs for short. The absolute best way to think about them is as master switches, or maybe dimmer knobs for your genes. They don't change your DNA itself, but they control which genes get turned on or off. They're what tell a cell, hey, you're a skin cell, or you're a liver cell. And as it turns out, they also control how a cell behaves as it gets older. Finding the right one? Well, that's the key.
Okay, so the big question, right? Did this fancy search engine actually work? Well, after running the platform on aging human skin cells, the researchers didn't just find one, they found several incredibly promising master switches. Yeah, it worked.
And here are the top 4 that they confirmed. Check this out. For 2 of them, E2F3 and EZH2, the secret was to turn them up, to basically crank up their volume. For the other 2, STAT3 and ZFX, the magic happened when they turned them down or quieted them. It really is that simple, just like flipping a light switch up or down.
And the results they saw in the lab, they were just astounding. When they flipped just one of these switches, this whole cascade of rejuvenation kicked off. Old cells started dividing again. Their ability to clean up junk proteins got way better. Their cellular power plants, the mitochondria, got a huge boost. And that whole process of cellular aging, senescence, just slowed right down.
And here's the kicker. This is where you really see the power of this new approach. The researchers found that tweaking just one of these factors, just one, produced results that were pretty much on par with using that entire complex 4-factor Yamanaka cocktail. Think about that. One switch doing the work of 4. That is a massive leap forward in simplicity and potentially in safety too.
All right, but let's be real. What works in a petri dish is one thing. The real test, kind of the holy grail for this type of research, is seeing if it works in a living, breathing animal. So, naturally, they took that next massive step.
So the research makes this huge leap, right, from in vitro, that's cells in a dish, to in vivo, which means inside a living animal. The team decided to test one of their star players in the liver of an old mouse, since the liver is an organ that really shows its age.
And the experiment itself was just beautifully simple. They took one of their best switches, Easy's H2, and used a safe method to turn its activity up, but only in the liver of old mice. That's it. No complicated cocktails, no weird side effects. Just one single targeted tweak.
And the results? After only 3 weeks, this is where the story goes from just promising to, honestly, revolutionary.
First off, they saw more than 50% decrease in 2 of the classic signs of an aging liver. Those fatty deposits, that's called steatosis, and scarring, which is fibrosis. I mean, the actual physical damage of aging was being reversed. A lot.
But it wasn't just skin deep, you know? The liver's actual function improved dramatically. These old mice could suddenly control their blood sugar again, just like they were young. Their glucose control was restored to the same levels you'd see in a young, healthy mouse. That one single switch brought back youthful metabolism. Incredible.
So let's zoom out for a second. What's the big picture here? Why does this one study matter so much? Well, because it really signals the start of a whole new era for how we even think about aging research.
And hey, don't just take my word for it. The researchers themselves said, our work expanded the list of candidate rejuvenating TFs for future translation. What that means is they haven't just found one key to one door. They've shown there are a bunch of keys, and more importantly, they've built the machine that can find them all.
And honestly, that is the most important thing to take away from all of this. Finding EZH2 and those other factors, totally amazing. But the real breakthrough, the thing that's going to last, it's the platform. It's the search engine itself. This is a tool that scientists can now point at any cell type, any tissue, any age-related disease, and just go hunting for these simple, powerful master switches. And that leaves us with a pretty incredible thought, doesn't it? For the first time, we have a real, repeatable way to find the keys to rejuvenation. It's like we've just been handed a treasure map. The only question now is, where do we start digging?